Good morning. First of all, I would like to thank the organizing committee by the nice invitation. So I'm going to share my presentation now. So I'm from Lisbon. You are all welcome to visit us. We are a school of engineering, including biomedical engineering, where I'm coordinating a master degree on that area. So today I'm going to speak about precision medicine and serum metabolomics to achieve that precision medicine. So what is precision medicine? Actually, precision medicine implies that we need to characterize mechanistically the diseases, try to understand the mechanismic associated to all subtypes of diseases. Usually we don't have a defined type of disease, the subtypes. And if you understand mechanistically these diseases, we could deliver more precise medicine. We could conduct more precise diagnostics and more precise therapies. A classic example of this is the breast cancer, for instance. We have several types of breast cancer. And if you understand what kind of type of breast cancer, for instance, if is overexpression hormonal receptors, then the best therapy would be a therapy that will down-regulate these hormonal receptors, and so on and so on. So there's a lot of examples today on medicine. And to define these mechanistical diseases, we want to discover these biomarkers that will define the different types of diseases. And my goal is discovering these biomarkers. So a biomarker is a quantity. We need to quantify it and associate this characteristic to a disease's phenotype if you are discovering biomarkers for diagnostic. And the more efficient mode to discover these biomarkers is if you are able to analyze systems, the whole systems, considering a subtype of molecules. For instance, technological speaking, if you are looking for the genes, if you want to achieve almost all the genes in a system, we call it genomics. So, and technologically, we have evolved a lot in the last 20 years. For instance, 20 years ago, when we achieved the first draft of a human genome, we took more than 10 years, and humankind speaking, this cost us around billions of dollars. So today, 20 years after, we are achieving almost the same result, or with a more sensitive and specific analysis, even so, with much cheaper. So this is an example how technologically we have evolved, and we are able to analyze for a given system one type of molecules. But instead of speaking about genomes, if you want to understand what exactly the system is doing, and not the potential of the system, what we can do it, because all our cells have the same genome, but all our cells are doing different functions. So to understand what exactly the cell is doing, instead of looking to the genome, we could start looking what the cell is doing, like the metabolome or the proteome. So when we going down on the system, we are closer what the system is really doing. So I'm focused my research on metabolomics mostly. So looking for that, what kind of techniques exist to analyze the metabolites on a system, what the system is conducting. Well, all the flow on a metabolomic analysis is highly complex. We start to prepare our sample. For instance, if it's a serum sample, we need to extract first to eliminate all these macromolecules like genes, like RNAs, like proteins, to remain on the system, the small metabolites. So this is the first task. Then we need to think about the technique we are applying, And after we are going to get a huge quantity of data, and then we need to think about the bioinformatic tools to analyze all this huge quantity of data. So starting for the analytical technique, well, actually we have, technological speaking, different types of techniques. For instance, the most usual type is when we associate a chromatographic step to separate all these metabolites, and then a mass spectrometry to detect and then identify all these metabolites. But even so, if you choose these classic ones, chromatography, we need to decide that liquid, gas, and among the chromatographies, what kind of separation we will select on this kind of techniques. So there's a lot of different techniques, and all of them are analyzing subtypes of molecules, so subtypes, subsystems of the metabolome. And this is highly complex, so it's impossible to have the whole metabolome of the system, but you want to characterize the metabolome associated to a metabolic state of the systems to be able so to do these subtypes of diseases, so more precise diagnostics and more precise therapies. So one of the techniques that we could also apply, and is a much simpler one, despite is not so sensitive, is the one that I'm been working with, that is Fourier transform infrared spectroscopy. On this kind of technique, we are looking for vibrations on molecules between atoms. And we are not able most of the times to quantify a defined molecule, but even so, we are acquiring the whole molecular profile of the system. So we are acquiring a fingerprint of the molecular system at that particular time. With the advantage of the technique is very rapid, simple to apply, much cheaper, and applicable to large set of samples, so to high throughput analysis. I've been working this kind of techniques in different kind of systems. For instance, for a much simpler system here with Escherichia coli system producing plasmids, we were able to, from this one spectra, to infer the concentrations of different molecules. Even so, the quantity of cells present, the plasmid, the metabolites like glucose, lactate, acetate. This was conducted on microorganisms, but we are able also to apply this to human cell culture, in this case on stem cells with mesenchymal stem cells. And we are able to develop very nice models predicting all these molecules with a very high precision and accuracy. This is amazing. And looking at also to the spectra, we're able to decompose the spectra to predict the defined molecules, for instance, along the time of a culture of bacteria, on this case, to predict, for instance, apparent transcription level, so the level of the expression of genes, the metabolic status of the cells, the turnover metabolism, so the quantity of energy that the cell is being producing along the time. And all this gives you very nice clues about actually that this spectrum is associated with the metabolic state of our system. So the first example where one of the first examples, actually wasn't the first, we applied this is to predict the exposition of humans, professionals, to drugs that could give genotoxic effects. So we're going to alter DNA and could start carcinogenic process. So we look for volunteers. Most of them work on an hospital environment and were exposed to surfaces that were contaminated with drugs that can act as genotoxic. So they can affect the human genome, alter their human genome, so they are very toxic. And we were able to predict these abnormalities of the human genome on cells from the immune systems present on the blood. To observe this, the conventional mode is to extract blood, isolate the cells, grow the cells during almost 1 week. And after this, and after different process, we were analyzing at optical microscope the nucleus of these cells to look for nuclear abnormalities, so genotoxic effects. This task takes lots of time. We need specialized personnel, and so is not conduct usually to monitoring this kind of events. So, and we want to analyze if the spectra from the serum, so this is a infrared spectra from the serum, and if the spectrum for around 40 professionals that present this in relation to 40 people that didn't present these genotoxic markers, and if the serum spectrum was also different from these two populations, and were. We found statistical different bands that were different in average on these two populations, the one that was exposed and the one that were not exposed. So more applying machine learning algorithms to these spectra, we were able to predict if this person presents genotoxic events. So we are able to observe that we are able to predict with a very high accuracy, around 0.86, so it's very high, with the high precision, 88%, and with a high sensitivity, also 86%, if that person were exposed and already present genotoxic events. With the advantage that this takes 2-3 hours to analyze in a very cheap and very sensitive and specific mode. So this result was very interesting. We also applied this to the immune cells. So instead of looking at the serum, we start to look at immune cells and understand what the immune cells are predominant in a certain type of disease is critical. For instance, in transplant human persons, people that receive transplanted as kidney, the mechanism of rejections is important to understand, because in function of the immune cells that are associated, the therapy is completely different. And again, we achieved to predict what kind of immune cells were activated based on the spectrum of these cells. And this is conducted very, very quick. In around 3 hours, we are getting the results with a very high accuracy and precision and sensitivity and specificity. And more directly, when applied this also to predict, for instance, in transplanted persons, the ones that we are developing rejection process of the kidney, we were also able to predict what persons were rejecting the kidney. So associating this to this, we could define the best therapy for this kind of patients. All of these were conducted with infrared spectroscopy. So, and now we are working another project, and we are also conducting the conventional mode to retrieve the metabolome, that is using a chromatography associated to mass spectrometry, where we were able to identify the metabolites in opposition to the infrared spectroscopy. And interesting, we were achieving very nice predicting models based on these two platforms, so based on MES, and based on infrared spectroscopy. So the models we are starting just now and the models we were developing are very nice. The last example that I'm going to present, because it's a slightly different platform, technological speaking, is a work that you actually I started working on this almost 20 years ago when I started on this area. And we want to know if the spectra from prostate cells were able to predict and diagnose prostate cancer. So we're working with non-healthy prostates. So we have hyperplasia, that is not cancer and cancer, but with some similarities when looking at cells sometimes, and difficult to discriminate it. And we'll look to average spectrum, and even so the average spectrum is slightly different. We're not statistical difference, so we are not able just looking for the spectrum as previously to identify the cancer. So we needed to look at the tissue. So we need to do analysis of the tissue, not one sample, but conduct the space visualization. We need to take a spectrum from all these different locations. So if you were a tissue sample from prostate, from a biopsy, for instance, we need to get all these spectra from all these spaces. So we are speak about imaging, and we are conducting this with a microscope that, as we call it, the focal plane array. So it's 64 to 64 detectors. So we're able to achieve more than 4,000 spectra distribute in small areas. And the microscope can conduct this in micrometers distance. So it's amazing equipment. We are retrieving hyperspectra, so huge quantities of spectra, and we want to understand if using this we're able to diagnose the cancer. Sorry. Just for a practical example, if we were able, these here are optical microscopic observations, and this is infrared reconstruction of this based on the area of some of this peak of infrared spectra. So if you integrated this peak, so the area, and if, for instance, this is proteins, we have the reconstruction of the distribution of the proteins concentration of this sample. That is for curiosity. But we want to understand if all these analyses we were able to achieve a diagnostic. So we made very complex data processing analysis. Not only we applied different techniques for the spectra pre-processing mode, but also reduce data and then evaluate different types of data processing, supervised and non-supervised algorithms. And we compare all these combinations, and by comparing all these combinations of pre-processing, processing data of all these spectra, we were enabled to achieve accuracy to predict the cancer, the prostate cancer, in 97%. So that is huge. So we compare with classic observations made by a physician, so a trained physician, very specialized physician. So we were very, very happy. So we conduct all this, and just to end my presentation, and this technological speaking and associated to very complex data analysis is one of the most information-rich and concise way to represent the whole of the system, because we are not looking only for the metabolites, but we are looking for also the genes, also from the proteins, RNA, and so on and so on. So this is my last slide, so it was a pleasure to be here. I hope that you enjoy my presentation to discover biomarkers from serum analysis, from cells analysis, from tissue analysis, to achieve a more efficient medicine. So thank you very much for the attention.